Okay, so welcome everybody. We are going to start today's seminars. Today we have Rafael Molina. He recently joined the EBD in January as a Ramon y Cajal researcher. He has been, he's originally from Seville, and he has been working also in Madrid, la Universidad Autónoma de Madrid. He has been also previously working in Switzerland. So he's going to talk to us about his work. He's a phylogenetic plant ecologist. I hope that you enjoy the talk. Rafael, welcome, and the floor is yours.
Thank you, Isabel. So, thank you everyone for showing up. It's really a pleasure to be here and to give this talk with so many familiar faces going back home. Just because maybe not all of you know about myself, I will briefly introduce my main research line before to go into the talk. So essentially, I work in three different lines. On the one hand, what I call assembly phylogenetics, which is ecophylogenetics, but applied to different spatial scales from the community to the macroecological scale. Also, I work developing phylogenetic methods to apply in ecology and evolution. And I have this third line, which is with which I am interested in understanding the links between biodiversity and human well-being. And more specifically, for well-being, I mean understanding how phylogenies can teach us about plant use patterns. Plant use patterns in terms of medicinal plant or any plant that we humans use. And all this research line has the phylogenies as a background. Although today I'm not talking about phylogenies, believe me. So if we think in a mechanism that is probably orbiting the mind of all ecologists nowadays, that is competitive exclusion, right? So these winners, they call paradigm, but we have different species with different competitive abilities, and only the stronger, or the more adapted ones are those persisting, and the weak species are excluded from the communities, right? But we also know that there are many species-rich communities out there, and this is especially highlighted by the paradox of the plankton by Huntington, who realized that there are actually many species out there. So there is an anomaly in biodiversity that we must explain. And there has been different explanations for this. For example, perturbations prevent communities to reach an equilibrium. So the rules of the game reset each time there is a new perturbation. We also have intransitive competition, right? When we don't have this hierarchical structure, but some species are stronger than others, but the stronger one for one species may not be the best competitor when it is about competing with a third species, and so on. And we also have facilitation, okay? We have been doing a lot of research about how facilitative interaction between organisms is. But there is a problem with this, especially applied to plants. My talk about today is about plants. So the problem is that nowadays, facility is seen as a transient mechanisms. When adult plants or nurse plants are facilitating recruits of other species, it's okay, we have this positive interaction, but the recruits eventually grow up. And when they grow up, they tend to compete. So, for example, in this meta-analysis, the author conclude that there is a strong fitness cost for nurse plants, implying that facilitation can temporarily, maybe in a transient manner, increase biodiversity, but eventually it will not hold. Eventually we will end up in competition again. And therefore, facilitation is not completely seen as a mechanism that can structure communities and biodiversity in the long term. And this is not the case with competition, which is the supreme force in ecology nowadays. Okay, so me and my collaborators argue that maybe we want to challenge this view of facilitation, just thinking in reciprocal facilitation rather than facilitation itself. So, for example, here we have two individuals of two different species, They are competing with each other. This is the orange arrows, okay? But the adult individuals are facilitating the recruitment of the offspring of the competitive species, okay? So if this facilitative interaction is stronger than the competition interactions, at the end of the day we have a net effect between the two species, and they could possibly coexist. So the rule or the requirement, the condition will be that facilitative interaction is stronger than competition, and facilitation, they must be reciprocal, okay? This is what we are proposing. They can be directly reciprocal, and as in this case here, or they can be indirectly reciprocal. In this case, for example, this individual, this recruit, which is a recruit of this species, this other species is helping this recruit of this one, and this one is helping the offspring of this third species. So there is an indirect effect here represented by these dotted arrows, which are driven by an intermediary species. Okay, here we have an indirect reciprocal facilitation. Okay, this is our theoretical background, our starting point. And in order to test this, to see if this reciprocal facilitation exists, we draw from this dataset, which is a global database of plant recruitment networks. Specifically, we count on 96 woody plant recruitment networks. We will talk a lot about this in the following minutes. They span 23 countries across 5 continents, and totaling 1,326 species. This is a recruitment network. This database is available, readily available for free as a data paper, so everyone who is interested can actually use this. Okay, and as I said, this was a global effort to sample these recruitment networks. Essentially, what we did, we went to the field, and within a given area, we sample all species growing there. And for each species growing there, we count all the recruits, all the small saplings, okay, of every species. And we also take note if these recruits are growing in open space or under the canopy of which species. So we have all the recruits, and we know where these recruits are growing up, in open areas or under the canopy of another species. And this is essentially, from that we can construct a recruitment network, as we see right now. So this is simply a map of the sampling points. And this is how we construct the network. So we have these arrows, right? These arrows are representing a significant positive interaction, recruitment interaction. And those interactions are inferred from a significant co-occurrence pattern between the recruit of a given species and the canopy of another species. So we have the canopy cover, in this case of species E, in the sampling area, and we have PT. And we know where these guys are growing. So if these guys are predominantly growing under the canopy of this species, we can conduct a chi-square test to see if there is a significant aggregation of the recruit under the canopy. We have a significant positive interaction. Okay, and this is our evidence that this species is facilitating the recruitment of these other species. We can later discuss about whether this inference is reasonable or not. We also can have random pattern, as in this case, and we can also have spatial segregation. In some cases, we find that the recruits of a given species seems to avoid the canopy of another species. So we have positive, neutral, and negative interaction, recruitment interactions. And this is just a theoretical example, just to show different things that we analyze in this network. So we have each point is dot here is a population composed of adult individuals and also the recruits individuals, and the arrows are facilitative interactions. Gray ones are facilitations that are not reciprocal. is a facilitation event that is not returning to the benefactor. But the arrows in colors are reciprocal interactions. So this interaction here, this facilitation event from this species to this species is returning to the benefactor eventually, following different pathways. In this case, for this research, we focus on the shortest loop reciprocating each facilitation event. Okay, for example, in this case here, this facilitation event is reciprocated through this pathway. It is also reciprocated through this other pathway, but we focus here. Why so? Because we think that these shortest loops are less likely to be disrupted. So we think that there is a higher probability that they are not disrupted, and therefore they are more. They will be more predictable. They will be more stable in time. Let's say. Okay, that's why we focus on the shorter loops for all the analysis we conduct here. We can also later discuss why or what are the implications of this procedure. Okay. Well, I think we can go. So first, what we thought we did was describing the patterns of reciprocal facilitation. In this case, we count the number of population that acted in our committees only as beneficiaries, only as benefactors. Both as beneficiaries and benefactors are non-facilitating anyone, or neither being facilitated. And we found that in most cases, most population acted both as beneficiaries and benefactors. And also, in most cases, those facilitations were reciprocal. That's what we found at the population level. If we see the data set at the interaction level, we found that of all these reciprocal interaction, most of them were indirect facilitation, mediated by one intermediary species. We have this direct interaction, this amount of direct interaction, only two species involved. But in most cases, we have one or two intermediary species. Okay, that was the pattern emerging after exploring the data set at the interaction level. Okay, population level, interaction level. And finally, what happened at the community level. So at the community level, we found that 58% of the community showed at least one reciprocal facilitation event. Okay, and we also compute the degree of reciprocity, which is the number of the frequency of reciprocal facilitation event compared to all facilitation events in the community. And on average, we found 51% of degree of reciprocity, considering all the communities. Okay, so this pattern suggests that reciprocal facilitation seems pretty prevalent, at least when we seek facilitation in the recruitment. Okay, keep in mind that we are talking about facilitative interaction on the recruitment. Okay, it's not between adults individuals, on the recruitment. It seems pretty prevalent. Okay, and this is very nice, but this is just a picture. It's a static picture. Maybe this is a picture, and after a few years it will disappear. So we don't know if in the long term. Okay, so we start to think, okay, what mechanisms will explain the long-term persistence of this facilitative recruiting interaction, direct and indirect facilitative interaction? So first, we think that there must be a condition that is that the shortest facilitation loop, reciprocal facilitation event, the stronger the facilitation event must be. Because if we have long loops like this one, if one species is not facilitating the side to interrupt the facilitation chain, the whole castle will burn down. So as these loops are longer, we expect stronger interactions. Okay, that's condition one. And we also envision a condition two, which is related to the negative interactions. Okay, so the idea is that we shouldn't observe negative interaction within the loops, or we should observe a slow frequency of such negative recruitment interaction within the facilitation loops, because those negative interactions will force the loops, will doom the loops to the extinction, right? It will be breakdown. So if we have these interactions here, the red arrows now are reflecting this negative interaction. We are actually not expecting this, but something like this. We expect negative interaction in the recruitment, but only out of the loops, of the facilitation loops. Okay? Does it make sense to you? Okay, how we measure this? So to measure condition 1, to test the condition 1, we took each population as an observation, okay? And we essentially relate the number of intermediary species reciprocating the facilitation event provided for by each species against the facilitation, the strength of the facilitative interaction in the loop, okay? So for example, if we have this species with 3 facilitation events. First, we find the shorter loops reciprocating each facilitation event, and we count the number of intermediary species, and we do an average. That will be our explanatory variable. And the dependent variable is the strength of the interactions reciprocating the loop. In this case, if we have these 3 facilitation events, we have one interaction here, and the shorter loops for this event, there are actually 2. They have the same length, 4. We do the mean between the 2. We have this strength here, also this facilitation event, so we compute the facilitation event, the strength of this interaction, and this interaction here, okay? Using this index, the nurse intensity index, which essentially is higher, meaning a stronger facilitation interaction, if we have a low canopy cover of the benefactor, a high number of recruit individuals of the beneficiary growing beneath that canopy, and many very low recruits of the beneficiary species growing in pervasive open gaps. This is the idea of the index, how the index works. And this is the hypothesis. We are relating the number of intermediate species in the shortest loops, reciprocating the benefits, and the average strength of the facilitation returning the benefits. And for condition 2, maybe it's more simple. We calculated the frequency of negative interaction within the shorter facilitation loops, reciprocating the benefits for each species, and we compared that frequency of negative interaction within loops to the frequency of negative interaction across the whole community, the whole network, okay? And we expect this probability to be, or this frequency, if you want, to be lower than this one. And we test that using Wilkinson pair test. So results. Concerning the first condition, we found this positive interaction with a substantial goodness of fit. And concerning condition 2, as represented here, we found a lower probability of negative recruiting interaction within this loop than elsewhere in the community. So we found empirical support for our 2 conditions, exploring the data. What is the conclusion? The conclusion is that this high prevalence of indirect reciprocal facilitation is supported by the 2 mechanisms. So the 2 mechanisms are supported. It seems that they are not transitory. So instead, we propose that if these conditions are given, and we are proving that they are given, at least in our data, there will be the material for natural selection to assemble this reciprocal and to be persistent in the long term. And this somehow mirrors the ideas of Trevers in the 70s about the reciprocal altruism, okay, which was maybe abandoned a long time ago, not very popular, not as popular as competitive exclusion, but it was proposed that reciprocal altruism between populations could be a way to explain high biodiversity levels out there. And also, if we have this reciprocal facilitation, prevalent reciprocal facilitation, and if we have these two conditions, we have proven these two conditions explaining probably explain the long-term persistence. We also thought that maybe indirect reciprocity is also explaining the species richness. Okay, it makes sense. So we evaluated whether the degree of indirect reciprocity in each community is related to species richness, and we found a positive relationship as expected. And we actually tortured this data, just taking into account the extent of the sampling methodology, because the data set was assembled using 3 different protocols, current and past climate, network connectance, and many other things. Also the type of biome and different things, just to test if there is an artifact. We didn't find any artifact, so the relationship persisted. Okay, so we are very enthusiastic at this point, but still not completely satisfied, because we still have a static picture. We are missing the temporal arrow. We are missing the temporal line. This reciprocal facilitation is happening, but we don't know if once these interactions occur and once these recruits are growing happily under the canopy of another species, maybe they are not able to reproduce, for example, for any reasons. We don't have a measure of fitness, and fitness is very complicated to measure, as you probably know, especially if we are talking at the global scale. So what we did is to complement this analysis with a modelization exercise using a load-coupled Volterra model. I will try not to focus much on the details, just to go quickly, but to provide enough information for you to understand what we did here. So as you know, with this model we can model, sorry for the redundancy, the population dynamic of every species, taking into account the intrinsic growth rate of the species, this parameter here, and of course the abundance of the species for which we want to measure the population dynamic, and the abundance of any other species in the community, and this pattern here, okay, which is the interaction, the net effect of the interaction between each species in the community and our target species. And this parameter here is computed from the facilitation and competition interaction matrices. And this will be the backbone of our model, okay, concerning intrinsic growth rate, rather than using, well, these are actually sphericals without no friction, as Violeta's cows. Here you are. But just simply, just try to give more realistic, to bring more realism to our model, we compute the intrinsic growth rate for each species as the fraction between the number of recruits growing in open areas divided by the area in the community in open gaps, representing open gaps. This will be our intrinsic growth rate. We can discuss, but we think that it's better to compute this, to use this, rather than simply modeling intrinsic growth rate, measured as here. And what we did was sampling from a uniform distribution bound between the confidence levels of this sampling from here, using a uniform distribution. And these are the values for this parameter that we are using to fit our model. And what about facilitation? In our model, not all species in the community will be able to facilitate any other species. Only the species that we are observing in the network facilitating another species, they will present a positive coefficient here. Otherwise, it will be 0. Okay? And however, for competition, now we can simulate. So in our simulation, all species in the community will be able to compete with other species. And this probability will be pretty high, 0.95. Okay, so there is a high probability that our species are competing with each other. to some extent. To what extent? We will see. So when we have the two matrices, we have the interaction coefficients, the net interaction coefficient, but we have to fill in these matrices. Okay? How? Well, depending of the scenario. We test 4 different scenarios. Scenario 1 is simply the topology of the networks. Okay? So we are sampling from the empirical distribution of Nurse intensity index, as here, using again a uniform distribution, bound, and we are sampling the values, the interaction, the facilitation and competition values from this distribution. Okay? And this is how we are filling here. Competition, we fill there with a 0.95 probability, and facilitation, we fill with a value if observed in the real community, in the topology of the network. Okay? This is a scenario 1. A scenario 2. For a scenario 2, we will have some interaction that on average will be higher, stronger. Okay? And the interaction will depend on the number of intermediary species. So if we have a facilitation event, for example this one, and the 2 species involved form part of a shortest loop, reciprocating the event, then this interaction will be stronger, and it will be more strong as the number of intermediate species is high. Okay, this is what we are simulating here. Therefore, in a scenario 2, some facilitation interaction will be stronger if the 2 species interacting are part of a facilitation loop, a shorter facilitation loop, reciprocating the given facilitation event. This pattern here, which is the slope of the relationship between these 2 terms, is sampling randomly from this normal truncated distribution between 0 and infinite, and is centered in 0.075. We test different values for this to center this distribution. We found consistent results and realistic results. The idea was obtaining interaction parameters that are realistic, because we have the empirical values. Now we are simulating, and this choose is constrained for or is set simply to avoid weird values, weird unrealistic values. And still we use this one 0.075, but we test different values, and the simulations remains very similar, the result, the output of the simulations. And this term here, which is the intercept of the relationship, is simply the mean of the empirical distribution of interaction strength measured as the nursing intensity index from the empirical data. Okay? So if we have a 2 species involved in a facilitation event and there is no intermediary species, the term of the facilitation will be simply the mean. And for competition, we are simply sampling the values from the empirical distribution of the index. Then we have a scenario 3. In this case, we are playing with this term here. And in this case, some species, some pairs of species will show a lower probability of interacting negatively. They will show this lower probability when the 2 species are involved within one of these reciprocal loops that we have been talking about the few minutes before. Okay, 0.88. Again, we try different values here, similar results. And for facilitation, simply as in a scenario 1. Okay. And finally, we have a scenario 4, which is a combination of a scenario 2 and a scenario 3. So now we are combining our 2 conditions in the simulations. Just a summary of the simulation set, we have 25 replicates, 4 scenarios, 96 communities in total, and this total number of simulations, 100 time steps per simulation, and the response variable is the survival fraction of the species per community at the end of the simulations. We use this output here as our response variable as a function of the degree of indirect reciprocity in our communities in interaction with this scenario. This is the model. And this is the output of the model. So in a scenario, using a scenario 1, the raw facilitation network, there is no relationship. When we consider a scenario, some interactions, facilitative interactions are stronger because they show many intermediate species. There is a trend to a positive relationship, but not really striking. In a scenario 3, we have also a positive trend, but not really striking. And again, when both iterations, both conditions are included in the model. In scenario 4, in that case, we found a strong positive relationship between indirect reciprocity and the proportion of survivor populations. We didn't measure fitness, but we simulate the outcome of the fitness along many generations. Well, yes, this is simply the conclusion is that our two conditions, which were empirically tested, are also supported by numerical simulations as a complement result of our research. And just coming to an end, so simply a few take-home messages. First, reciprocal facilitation in the recruitment seems highly prevalent, at least in the plant world. The data also suggests that this prevalence can be explained by two conditions, at least: a stronger iteration in longer facilitation loops and a lower probability to find negative interaction within these facilitation loops. And finally, we have greater diversity, more species richness in communities showing a higher degree of indirect reciprocal facilitation. And this result is coming from the dataset, is empirical, and is also support with our simulation toy, let's say. And to share this paper as a highly recommended reading, what was published in Nature American Naturalist a couple of years ago, 3 years ago, which entitled Moving Beyond the Diversity Paradox, the Limitation of Competition Based on Understanding the Species Diversity. This is a blah, blah, blah paper. They do some analysis also exploring how many papers cite facilitation versus competitive interaction in the literature during the 20th century, different things, but they want to grab your attention into this final line that I'm going to read literally. So the authors conclude that we argue that we must excavate the ideological foundation on which competition-based coexistence research has been built to move beyond the current perceived diversity paradox. To that end, we propose shifting the baseline expectation of coexistence research, introducing the notion of a coexistence principle, which positions the persistence of multi-species communities as the rule rather than the exception in nature. Maybe, and this is something that I give you as a challenge, or maybe something to trigger discussion, maybe it's time to move from trying to explain the anomaly of higher diversity of species-rich communities. Maybe that is not an anomaly if we consider that species can help each other reciprocally, as already envisioned by Robert Triggers in the 70s, and as we are showing in this research. And that will be all for today, at least from my part. Simply I want to thank my collaborators of this project, which are Miguel Verdú, Julio Alcántara, Alicia Montesinos, José Luis Garrido, Ezequiel Antorán, who is the PhD student of Joaquín Calatayud, more highlighted here because he is my second hand on this project, on this research. Okay, the stronger co-author of, and my favorite network ecologist, I must say. So thank you very much. I will be happy to respond to your questions.
So thank you, Rafael. So we have time for questions. Okay.
Super cool talk. Thank you so much. Do you have thoughts about the kind of mechanisms for why species would always be looking to the next neighbor for having higher recruitment? Is it they all need shade, but they want to be in a different soil type or disease? Like, is it kind of what do you think is the underlying reason why you would start to build up these loops?
Okay, that's a very nice question, because to this day we don't know. Maybe there are some traits there which are being selected for promoting this facilitative interaction. There is some literature, narrative literature, just talking about the need to explore this avenue, but so far we have no idea. I mean, we don't know if there are, for example, certain phenotypes promoting this recruiting interaction. For sure, if a plant has a canopy which produces a shadow environment in a desert, for example, probably that is a trait, phenotypic trait, that is being selected, let's say, for maintaining this reciprocal facilitation loop in that specific habitat. But maybe there are other traits. Also, maybe there are also phenotypic traits explaining this tend of recruit to grow or to seek for the canopies. But so far, we have no clue, almost no clue, about what are the phenotypic mechanisms behind these aggregations.
Thank you very much for the talk. I have a kind of follow-up question of Lauren, and is that your results are based on facilitation loops, but have you thought about including some null model in the definition of your indexes, especially the nursery index, in the sense that I would really like to see how in the simulations, how the systems evolve with the same amount of facilitation, but distributed without loops. Because I think that as the key are the loops, maybe you should just compare it with a null model dropping out those loops.
Mm-hmm.
Yeah, well, it's a suggestion, not a question, sorry, but
yeah.
I was just curious about whether you thought about that.
Well, what we have done is exploring whether the degree of reciprocity is simply not an artifact of having more species in the community. Because we have this positive relationship, and we thought, okay, maybe simply, if we have more species, maybe is more likely to have higher reciprocity. And we have conducted a null model analysis in order to assess that specific question, to remove that possible artifact. The results remain consistent, that there is not an effect on that way. And also, maybe it's not related to your question, but I forgot to mention, even there is empirical evidence in the literature that there are genuine canopy effects on the recruitment. This is something that has been empirically tested. You can have, in some cases, more than 10 degrees Celsius degrees beneath the canopy compared to the area surrounding. There is a reality that canopy effect can increase the fitness of the species. We have evidence of that. But one can think, okay, but maybe in some cases, your spatial co-occurrence between recruits and canopy species is simply because they share the same preference, the same niche preference, for example, right? So what we did is just to remove sequentially a certain fraction of the significant interaction from each community, 5% of the interaction, 10, 20, and up to 30% of the interaction, and reanalyze the data, assuming that those interactions are not due to genuine canopy-driven effects, but other effects. And the results persisted. This is also another character that we applied to the data, and still the pattern persisted.
Thank you.
Finally, thank you. So I think that the question about the mechanism underlying all this, it's quite pertinent, because perhaps you are familiar with the Greenberg gene theories, in which plants can recognize other plants depending on the genotype, and so there's a lot of things going on, that plants collaborate rather than compete, but this depends on the recognition of the genotypes. So once more here, the genetic diversity of these populations can be quite important. The more diversity, the more probabilities, the higher probabilities, to establish this kind of connections and recognitions between genotypes. But anyway, my question was a bit different, and I think an important gap here is the role that perturbations play in all these things. Because I've been thinking all the time in your talk about, for example, mixed forest in the Mediterranean.
Yes.
So after a fire, pines take over because they need sunshine, but oaks need shade, so they start to recruit under the pines. Over time, the situation flips upside down. In that case, the oaks thrive and pines get out just because that there are no openings. So the perturbation is needed, to keep this long-term balance in the relationship between species. So what's your opinion about the role of perturbations, disturbances in your system? So if you introduce a disturbance in your model, what kind of relationships would you expect in terms of the effects of all these interactions on the species diversity that you are interested in?
Well, that's a big question. I don't have a certain answer, but what I can say, I didn't mention it. In this data set, we have communities which are in an early stage of the ecological succession, likely having suffering a perturbation. In an intermediate or highly developed or in a late successional stage. We did this. We asked the authors of the samplings to label their communities according to these classifications, and there is not an effect of the successional stage in the analysis, which is related to the perturbations. But maybe it could be interesting, for example, to explore this with the simulations. Probably will be a way to see if the pattern can recover after the perturbations, but we haven't tested this besides just the successional state, and probably there is an effect, but I can go further in this regard. And concerning your first question, it could be very interesting. For example, something that we didn't quantify here is how is the intra-specific variability on the interactions. Maybe, and actually this happened, the same species in a community in a site helping a given species, but in other different community is not helping, is neutral, or maybe even the opposite direction. So the same species could be a facilitator in a community and a suppressor of the recruitment in a different community. Why so? We don't have an idea for this. This could be also an avenue to explore intraspecific variability, probably making a connection with population genetics to see if there is a genetic signal there.
Well, my point was that this is not a physical question of shade and humidity. This is biological, right? That was my point.
Thank you, Rafa. It was very clear, the talk. Why didn't you use the negative interactions in your data set as competitive coefficients in your population dynamics model?
Actually, just simply to, we could have done that, definitely, but there are way less negative interaction than positive interaction in our data set, meaning that the empirical distribution is kind of funny, and that's the reason why we decide to use the empirical distribution of the nursing intensity index, simply because we have very few competitive interaction compared to the positive ones.
Thank you for your talk. I really enjoy it. And it was like a continuation about one of the questions, the one about the phenotypic traits that could. And I work with fleshy fruit species, and for me, your talk makes absolutely sense, because since species can share the same dispersal mechanism, it will make all the sense that they facilitate a species of other species of the same mechanism. Like behind a Pistacia lentiscus, you can find a lot of Philirea and I don't know, fleshy fruit species.
Yeah, because of the pellets of the bird.
Yeah, because there are more seeds arriving to that place.
Yeah.
And the recruits of the same species will be penalized, or maybe they are not. They have lower fitness because of Janssen-Connell or whatever.
Yeah, yeah, that's a very good question. Surprisingly, we didn't find this criticism from the reviewers, which is pretty evident with fleshy fruits. You know, we know that birds tend to select species just to rest there and to drop their thing there. So this will translate into an aggregation pattern. And for that very reason, among other reasons, we conduct this analysis of removing a certain amount of interactions, because and reanalyze the data in order to see if the pattern persists after removing those interactions, assuming that, for example, they came from species with fleshy fruits that are dispersed in the canopy simply because a bird was passing over and dropped the pellet there. We have some species with fleshy fruit dispersed by birds, but we also have many camephytes, lavanda, rosmarinus, thymus, a lot of them with fruit that are not dispersed by birds or any other animals in principle. So that could be a reason to explain spatial aggregation for some species, a few of the species in the dataset, but not for all of them.
No, sure.
Yeah, that's a very good question.
Well, welcome and thank you. And also for the presentation. I mean, I can see that you have been teaching at the university, so it was very well presented.
That's very true.
Yeah. A question is that, okay, this is at the earlier stage, and this might change with time when the plants are adult.
You mean
I mean
recruits?
that the interactions will change when the plants get adult.
Yes, exactly.
And is there, I mean, this data set will allow to see whether there are interactions that persist through time, and also at which point the interactions switch. Because these are seedlings, but what about the sapling stage? I mean, it's like we think about the seeds, the seedlings, and the adult plants, and so it's like, can you tell something about the life history?
Yeah, sure.
Effects on.
So they are saplings rather than seedlings, because we are talking about woody species. And this is exactly the problem. As I mentioned, maybe kind of loosely at the beginning of the talk, the current framework of facilitation does contemplate this shifting in the iteration from positive to negative as the recruits grow up. For example, this is illustrated by Oedipus, for example. As a kid, was okay. The kid was running there on the street of Greek, but when he became an adult, it was a mess. He started competing with each other, and finally he killed his own father, if I well remember. This is the idea, the general idea of facilitation. So some plants can facilitate the recruits of other plants, but when the offspring grow, we have a problem. They start competing to each other. And that's why the conclusion, for example, of this meta-analysis was like facilitation in the recruitment has strong fitness costs for the benefactors, and therefore facilitation can increase biodiversity in the communities, but temporarily as a transient mechanism. It will not hold in the long way is competition. And this is the view we are trying to challenge here, like, okay, maybe facilitation is reciprocal, we can fight as adults, but if we help the offspring of each other, the system can be, so even if we take into account those expected shifts in the interaction when the recruits grow up, this reciprocal facilitation in the recruitment can circumvent the issue and allow facilitation to be a mechanism promoting high species richness in the long term, along with competition. We are not negating the impact of competition. We are living in this world, and you can see competition in the field. We are simply saying that maybe facilitation has a more prevalent role than previously thought.
Thank you very much for your talk. I was wondering if in your recruitment network designs there exists the possibility to work with self-loops for the species that can facilitate or even indirectly negatively affect themselves?
Yes, we have a few, surprisingly, a few auto loops between a canopy individual and the recruits of its own species, either positive or negative, but very few compared to heterospecifics. This is something that that emerge from our dataset, the recruit net dataset. This is also something that I have observed in the field during my PhD, for example, when we did some field work, just watching what is behind the canopy of the species, and surprisingly, I found very few conspecifics recruiting under the canopy, which remind me of the Jansen-Collins hypothesis that reads that normally we expect recruits of the species away from the individual, from the source individual, because otherwise there will be competition, almost specific competition, and there will be an attractor for herbivores, for example. And that observation of Jansen fits to what we are finding here in the dataset. But yes, we have some auto links in the database, but maybe less than 0.75% or something like that. So very, very few.
I'm thinking about Violeta, the question that Violeta mentioned, because it might happen that in the data set, some communities might have only a few open areas, let's say. Could happen, no?
Yes.
So in those cases, the likelihood to find positive reciprocal interactions will be higher, but just because there is most area is under the canopy of other species.
But that's why we are applying the TS to each pairwise interactions. So for each pair of species in the community, we have the cover of species A and the number of recruits of species J. And we are testing whether the number of recruits of J growing under the canopy of A is higher or lower than expected for the given canopy cover of the canopy species and the number of open gaps. As you say, if we have a lot of canopy, we expect more of the recruits growing under the canopy, but then the chi-square test will be non-significant because the expectation will be that.
Yeah, but that, I agree, but the test will fail if the fraction of open areas compared to the fraction of areas under the canopy of other species are not more or less the same, right? Because you would have the sample size of each open areas or under the canopy would be very different in both cases. But it's a very technical question.
You mean if you have, can you say again?
Yes, I think the test would give you that answer.
Yes.
If the number of open areas is more or less the same as the number of non-open areas.
So if the surface in open areas is more or less
Yeah, because you said that you made a test comparing in how many open areas a species is present compared to in how many non-open areas the other species is present, right?
Yes.
If both number of open areas and number of non-open areas are very different, doesn't that test work properly?
I have to think a bit more about this, but well, essentially we have the number of recruits under the canopy of species A is higher than expected given the canopy of A. This is the test. So we don't know actually.
Okay, but you could also reshuffle the distribution of open areas and non-open areas.
Yeah.
And get the network of interactions, right?
Yeah.
As a null model.
Yeah, we could. Yeah, we could. Definitely. That would be a very interesting point to see, to check.
Okay, so it's time to finish. Thank you everybody for coming, and Rafael will be around, so if you have more questions, you can ask him directly. And thank you for coming, and have a good lunch. Enjoy.